In this lecture, we're going to talk about the eye and the ear, our two most complex sensory receptors. So let's start with the accessory eye structures, not part of the eye itself, but have a role in vision. So we have eyebrows and eyelashes, help shade the eye, help keep things out of the eye. We have eyelids, upper and lower eyelids. The eyelids blink reflexively to protect our eye and also to keep it moist. We have, notice this structure above the eye, this lacrimal gland, it's called. This is an exocrine gland with ducts that produces tears, lacrimal fluid, which lubricates and moistens the eye, and also has some bactericidal properties. It contains the enzyme lysozyme. So you're continually making lacrimal fluid. It washes across the anterior surface of your eye, and then it drains from the medial aspect through these lacrimal canals into the large nasolacrimal duct, which goes into your nasal cavity. So this explains why when you produce a lot of tears, for example when you cry, your nose gets runny. Okay, also in this picture you see this little bump in the medial part of the eye. That's called the caruncle, C-A-R-U-N-C-L-E. That contains some exocrine glands as well, sebaceous and sweat glands. So sometimes we see secretions of that in our eye, especially when we wake up in the morning. Also considered extrinsic or external accessory structures to the eye are the 6 extrinsic eye muscles. Extrinsic eye muscles. These are skeletal muscles. For example, here we see the superior rectus muscle. You do need to know the 6 for a lab. And they are systematically named. There's 4 of the 6 extrinsic eye muscles extend straight back, so they're called rectus, means straight. So there's a superior, there's an inferior, there's a lateral and a medial rectus, and then there are also 2 oblique muscles. So this view doesn't show all of those, but keep in mind that you do need to identify them. Their job is to move your eyeball around in the socket. So you might remember we talked about these briefly when we covered the cranial nerves, because cranial nerves 3, 4, and 6, that is their job, to innervate the extrinsic eye muscles. So you can use these to look up, look down, look right and left, roll your eyes, and they are important in vision because as objects move closer to your eye, you need to rotate your eyeballs medially, or conversely, as an object moves away from your eye, it becomes more distant, your eyeballs rotate laterally. So that's brought about by these extrinsic eye muscles. We call that convergence. Convergence is the name of that process. Okay, and then also another accessory structure, which is a little hard to see, is the conjunctiva, or conjunctiva, as some people call it. This is represented by this thin brown line layer, and this layer of this accessory structure is a mucous membrane. It's quite thin. It has blood vessels in it, so it's vascular. And part of it covers the white part of your eye when you're looking in the mirror. You see the white part of the eye, that's called the sclera. We'll talk about that shortly. That is covered by part of the conjunctiva. And then your eyelid, the inside of the eyelids are also lined with it. So even though part of it is on the surface of the eye, it is considered an accessory structure. So notice how if you follow it, there's this hairpin turn here. So this keeps things that get into your eye from going back further than the conjunctival bend here, fold. Okay, so those are the the main accessory structures of the eye. Now let's look at the eye itself. So there's a lot in this picture. I'm going to show you some simpler ones, but just to start with this, let's look at some of the major features here. So first of all, your eye is like somewhat like a hollow ball. There's a fair amount of space inside. So there's a very large cavity in the back of the eye. So this space back here contains a jelly-like fluid, so they call it the vitreous chamber, but it goes by other names, the posterior cavity, and it contains a jelly-like substance called vitreous humor, so it's like jelly. Unfortunately, we won't be dissecting the sheep eye any longer since our lab is now online, but if you were able to, you would have seen this when you cut open the eye, this blob of jelly, the vitreous humor. And then there's a smaller chamber in the front, the anterior cavity, which contains a much watery, a much more fluid, watery substance called the aqueous humor. So there are these chambers filled with various substances. There's the lens here in the eye. That's going to be important for helping to focus light rays on the part of the eye where the sensory receptors are, called the retina. So the actual sensory neurons, which remember that's the main thing a sensory receptor needs, are sensory neurons. So they are only in this thin yellow layer that isn't even in the front part of the eye. It's only in the posterior or 2/3 of the eye. That's the retina. And then there are some other layers which we're going to cover shortly of the eye. In fact, it's usually described in the back as having 3 tunics, 3 layers, and then as you get towards the front there are 2 layers. Okay, and then coming out of the back of the eye is the optic nerve, containing the axons carrying the action potentials away from the eye. So let's start by looking at the tunics, but I'm going to show you some pictures that don't have so much detail in them, so you can focus on the fibrous tunic. So this is the outer layer of your eye itself. Okay, so it's shown in light blue in this slide. So in the back of the eye, this outer layer it's a dense connective tissue. It's called the sclera, and it is opaque, meaning it's not transparent. It looks white, and you can see some of the sclera when you look in the mirror. I'll show you a picture later of this, but the sclera extends, up into here. So this is all the sclera of the eye. But then the anterior-most part of the outer tunic needs to be transparent, clear, so that light rays can enter, and that we call that the cornea. It's also a dense connective tissue, but the collagen fibers within it are arranged in such a way that overall it's a transparent covering. If this becomes cloudy, that causes vision problems. Okay, so that's a protective layer of the eye, and I meant to mention that it is avascular. It has no blood vessels in it. Now, the middle layer, however, obviously does. You can see that from its name. The vascular tunic has lots of blood vessels in it that are going to help feed the inner layer, which we'll get to next, the retina, and at least part of the outer layer of the eye. So we call that the vascular tunic. It's in the back. It's called the choroid layer, the choroid, and it, in addition to blood vessels, it also has a lot of melanin in it. Melanin, not for vision itself, but to absorb scattered light rays and enter the eye. So it's in a slide later that I'm going to show you. of the eye, the back of the eye, you'll see the choroid layer is very dark because of all the melanin. Okay, and then as you go forward, as you go anteriorly, the choroid becomes the ciliary body, and the ciliary body is in this area right here. The ciliary body has, I would say, two main functions. It makes the aqueous humor that's in this anterior cavity, and it makes it like we make most of our body fluids from blood, from the plasma. So it's a plasma derived substance that you are constantly making and draining away from the anterior cavity. So the ciliary body helps makes the aqueous humor. And then its other function is, if you look closely, you see these little lines running from the ciliary body to the lens here. Those are called suspensory ligaments, and they attach from the ciliary body to the lens. I'm going to show you a close-up of that in a moment. And what the ciliary body has smooth muscle in it that contracts and relaxes, pulls on these strings, and helps change the shape of your lens. So we'll talk more about that a little bit later. So controlling the shape of the lens is a big role of the ciliary body. And then as you move even more anteriorly, you get to the third part of the vascular tunic, the iris. And notice that the iris, in the middle of it, there's an opening. That's the pupil. So the pupil, it's depending on the size of your pupil, that determines how much light can enter your eye. Because when light rays enter the eye, they have to go through the cornea, the transparent cornea, through the aqueous humor, and then they can't go through the iris tissue itself. They can only enter through the pupil, and then through the lens, and then into the jelly. So the iris is, like the ciliary body, primarily smooth muscle, and it has by contracting and relaxing those muscles, you can make your pupil bigger or smaller. So, you know, your pupil can dilate or constrict. So if it's really bright out, you're going to constrict your pupils. If you're in a dark, say you walk into a movie theater, and you're going to have to dilate your pupils to let more light in. So that's the vascular tunic: choroid, ciliary body, and the iris. We'll look at it more closely soon. And then here's the inner layer, the retina. This is, I guess you could say, the most important part of the eye, and that this is where the photoreceptors are that respond to light with nerve impulses that then can be carried out the back of the eye through the optic nerve. So that's called the nervous tunic, or most people just refer to it as the retina. Okay, and then here's the anterior cavity in purple, just so you can see a little better. Again, what's in there? Aqueous humor made by the ciliary body. You're always making it and draining it away. You see this little canal here, that's where you drain away aqueous humor. It's very important that you be able to maintain the proper amount of aqueous humor in the eye, because if you have too much, it can put pressure on the lens and then on the jelly and back here in this posterior cavity, and then that can compress the retina and cause damage to the retina. So that's what happens in glaucoma. You have too much aqueous humor here, you're not draining it away properly, so usually they give you some medication, some eye drops to decrease the amount of aqueous humor produced. Sometimes they'll do surgery, not usually, but to make it easier for you to drain aqueous humor away from the eye as it's made. Okay, and then here we see the vitreous chamber, which is much bigger. This is where that jelly-like material is, the vitreous humor. Remember, humor is an old anatomy term for body fluid. The vitreous humor inside is quite different from the aqueous humor. It's not derived from your blood. It is not replaced routinely. In fact, it's made when you're an embryo, and then that's it. That's the vitreous humor you have. So it's not, replenished. There are some phagocytic cells in there that help remove debris. And one of the most important functions of this jelly like humor is that it holds the retina, which in this picture would be this layer that I'm coloring in red. It holds the retina against the rest of the eye, because your retina is actually not attached to the underlying choroid of the eye. It is just held flush against it by the pressure from the vitreous humor. The only place the retina is attached is right here, where the optic nerve leaves. That's the only place. So that's the vitreous humor. Okay, and then this is how you're used to, looking at eyes, I'm sure. So just to look at this for a moment, here we see the sclera would be this white part of the eye, but remember that it is avascular, so these blood vessels you see in here, those are actually in the conjunctiva, covering the sclera. So your conjunctiva comes over, and it does not cover the iris, it stops here. But any blood vessels you see when your eyes look bloodshot, those are in the conjunctival layer, not in the sclera. And then you see this ring, of this colored ring around the pupil. The pupil is that hole. That's your iris. And like the choroid of the eye, it has melanin in it that absorbs light, and depending on the amount and the variety of melanin that you inherit from your parents, that really determines your eye color. Whether your eyes are blue or green or brown or black has to do with the amount and the type of melanin. Okay, so one other thing that's hard to see here, but it's here, would be covering your iris would be the cornea. And because it's clear, we don't really see it as a separate layer, but there is that layer, the outer layer of the eye, sclera and cornea. This shows the iris, the muscles in it. So notice how these are smooth muscles. So this is not something that you control voluntarily like you do your skeletal muscles. But notice how the smooth muscle cells are arranged in different ways. So you have the circular layer and you have the radial layer. And so, depending on which ones you contract, you can constrict your pupil as you would in bright light, or you can dilate it in dim light. Okay, so that is the role, as we were saying earlier, of the iris, is to control the size of the pupil. Now, we aren't going to go into the physics of this very much in our lecture, but it is important to know that when light, which you can think of as waves of photons, when they pass through different media, light rays, they're bent. That's called refraction. So like when light goes from air through water, it's going to bend. So that's what they're showing you here, the refraction of light rays. Okay, so this has important implications for the eye, because in your eye, we talked about how your photoreceptors are in the retina of the eye. But the retina, and we'll go into this in more detail, the retina is not all the same. There is a region of your retina where you have a large number of photoreceptors, a type of photoreceptor called a cone cell. And those are highly concentrated in this region right here. It's not labeled, so I'll write it, called the fovea, or fovea centralis. So when you're looking at objects, you need to focus the light from those objects onto your fovea. The light rays need to focus on the fovea of the eye. And yet when the light rays pass through the different layers of the eye, each one is going to bend it. So let's follow a light ray here. So it's going to go through the cornea, which is going to cause refraction, through the aqueous humor, which will refract it even more, through the lens, which is a transparent, has a lot of protein in it called crystalline that crystallizes into a clear lens. It's going to bend the light. Then you're going to go through the vitreous humor, the jelly like stuff, which is all of this is going to refract the light rays. And so how are you going to get them to how are you going to direct them to the fovea part of your eye? And the main answer to that is that we're going to one of these layers that I just mentioned that it has to go through, we're able to modulate, to change, so that we can focus on light rays, much like you can focus a camera. So you can't change your cornea and your aqueous humor and your vitreous humor, but we can change the shape of our lens. So we were talking about this a little earlier, how our ciliary body has little suspensory ligaments that impinge on the lens, and they can become tighter or more slack, and thus you can make your lens, you can flatten it, or as you need to do when you're viewing far away objects, you want your lens to be flatter. As you look at closer objects, or as the same object moves closer, your lens needs to be more bulged. So your ciliary body is going to change that shape for you. Here's what that process is called: accommodation. So our ability to change the shape of our lens is a function of the ciliary body, the ciliary muscles in there pulling on these suspensory ligaments. And so you do that so that you can focus the light on the fovea. So here's a close-up of that, of the accommodation. So here's the ciliary muscle in your ciliary body. Here's the little white string, so those are the suspensory ligaments inserting on the lens. Okay, and the lens does have it has elasticity to it, which allows it to be somewhat malleable. Now this tends to decrease, of course, as you get older. So, people tend to have a harder time focusing on objects as they get older. So often, they need corrective lenses, because if you're not able to focus the light, maybe you focus it too soon, in which case you're referred to as nearsighted. So you would need some kind of corrective lenses for that, glasses, contact lenses, surgery, or you may focus the light rays too far, in which case it's called farsighted. In either case, you need some corrective lenses to help you converge on the fovea. Okay, so let's look at the retina in more detail. In this, going back to this complicated looking picture of the eye, I just want to orient you on the retina. Let's look at the retina. Notice it stops, or starts, whichever way you want to look, here, right here. In other words, if you go anteriorly from this red mark, there is no retina. As you go back posteriorly, the retina begins. And that anterior border of the retina is called the ora serrata, sort of a serrated edge. That's where the retina begins and goes posteriorly. Okay, also in this picture, if you can find it, here's the fovea, the central fovea, where you have to, macroscopically, it doesn't look like much. This is something you would need to look under a microscope to see. But That's where it's located. So that's where you need to change the shape of your lens so you can focus the light there. If you can't, then things look blurry and you're going to have a hard time, especially reading things like fine print. Also, I wanted to draw your attention to this area here called the optic disc. This is the significant in a couple reasons. This is where I was saying earlier your retina is attached to the rest of the eye, the only place. It's also where the optic nerve leaves the eye, and it's called the blind spot because you have no photoreceptors there. There aren't any. So any light that comes into the eye and falls upon this region is not going to be translated into nerve impulses. You are not going to see that image. Okay, now these are the two main types of photoreceptors. They're called rods and cones. They're both in the retina, again not equally distributed, but they're both there, and these are your actual photoreceptors because they respond to photons with graded potentials that may lead to action potentials. So we talked about these a little bit in our earlier lecture on sensory receptors, and we discussed how if for a sensory neuron, your stimulus is whatever opens your ion gates to cause graded and then potentially action potentials. So in the case of rods and cones, it's photons. For other sensory receptors, it's things like chemicals or mechanical pressure, but here it's light, photons. So these, we'll see shortly, there are other neurons in your eye, in the retina, but these are the only two that can directly respond to light with nerve impulses. So these are our actual photoreceptors. You can see they're from the picture, they're bipolar neurons. Remember, bipolar means you have two processes coming off the cell body. So these are both bipolar. They both contain photopigments, they're called. So they're kind of stacked like dinner plates in the neuron. They're made of a combination of a protein and some pigment. We call them photopigments, which are derived from vitamin A. So that's why we need vitamin A for good vision in things like that have carotene in them, like carrots and sweet potatoes. So these discs or photopigments in the eye are important because they are the link between the light hitting them and then opening ion gates to cause the action, or graded potentials and action potentials. So they have a very important role in how the photoreceptors work. So they both have them, rods and cones, but they are different from each other, and this leads to their different functions. Rods are the ones you use for dim light vision. So, in a room that's mostly dark, you see things in various shades of gray, or you don't really see in bright color, you can't see in detail. That's when you're using your rods, and they're also involved in peripheral vision. Cones, on the other hand, are for keen color vision. So they're the types of photopigments they have allow us to see in color. And there's 3 different versions of cones that allow us to see in different colors. So people who are colorblind, their cones lack one or more of these special types of pigments. So cones are the ones you're going to use to read or do anything that requires detailed eyesight. If you're painting or really anything that requires you to see clearly, they're the ones you would use in bright, during the day when you're seeing things in various colors. So those are in your retina. Okay, so here's how they're organized in the retina. The retina itself actually has two layers. It has a pigmented layer, which is shown here. This is a little hard to tell, but let's look down here. Here's the well, let me show it down here. Here's the pigmented layer. So it's right up next to the choroid layer of the eye. Okay, and it's actually not nervous tissue. These are epithelial cells, but they help process the precursors to the photopigments and provide them to the neural layer where the actual photoreceptors are. So if you look closely up here, you see the rods and cones are closely associated with the pigmented layer. Okay, and they need to be. Because, like I say, they're getting the precursors for making their photopigments from the pigmented layer. So if they become detached from it, that's a serious, eye disorder, or detached retina. So that's where the rods and cones are. Now, when light comes into the eye and hits these photoreceptors, that can cause action potentials that will travel in this direction. This is the direction of the nerve impulses. But notice that they're not very long. They're axons, and so they're not actually taking the action potentials to your brain. Instead, they're going to synapse with a middle layer of cells called bipolar cells, which are bipolar sensory neurons, but not photoreceptors. They themselves cannot respond to light. They are just synapsing with the actual photoreceptors. And then they in turn synapse with the ganglion cells. And it is their axons that form your optic nerve. So their axons are quite long. If you look at the optic nerve down here, these lines you see in there are the axons of the ganglion cells. So you've got the photoreceptor layer up near the pigmented layer, the bipolar cells in the middle, and the ganglion cells forming the optic nerve. There are some other neurons in here, but these are the three, or well, if we lump the photoreceptors together, these are the three layers that you need to know for your lecture and lab tests. Here's what it looks like under the microscope. So we see the sclera on the outside, and then the choroid with the see the dark pigments in there, that'd be melanin. Here's the pigmented layer of the retina, and then here we see the layers that we were talking about, the photoreceptor rods and cones. So here we see their cell bodies in this dark purple layer. Here are the bipolar cell bodies, and then here are the ganglion cells. And then again, their axons are forming the actual optic nerve that's going to take the action potential away from the eye. Okay, so this picture, this slide shows the pathway, what they call the visual pathway. This is the path that the action potentials take from the eye to their ultimate destination, which is going to be your cortex, your cerebral cortex, where the nerve impulses need to go to be processed. So here we'll just follow it along. So from the optic nerve, the impulses leave your eye, they cross over at the optic chiasm, or most of them anyway, and then they continue on along the optic tract. We're calling it optic tract now because technically we're in the central nervous system, where you call bundles of axons tracts, not nerves. Nerves are used in the peripheral nervous system. So your optic nerves are considered to be peripheral, part of the peripheral nervous system. In fact, that's cranial nerves number 2. And then the optic tracts take the impulses to the thalamus, which, remember when we studied the brain, the thalamus receives sensory input and directs it to the correct region of the cerebral cortex, although not for smell. But here it's going to the thalamus and then to your primary visual cortex of the cerebrum, which is back here in the occipital lobe. And that is where interneurons will process that information. That is something that we do not go into in our course, but that's where the processing goes of all this information that's arriving that gives us our conscious sense of vision. Okay, so that's the ear, or the eye, and now moving on to the ear, which is equally complex. Usually the ear is approached by dividing it into external, middle, and inner ear. So we'll follow that. But first let's just establish what sound waves are. That's what's going to be, that's the stimulus, and that's going to, for hearing, although your ear is actually a double sensory receptor, it's also for balance. We're going to talk mostly about hearing, that part of the ear, but I will mention the balance receptors in the ear. But let's start with hearing itself. So sound waves are waves of high and low pressure vibrations traveling through some mediums. You can't have sound waves traveling through a vacuum. It requires an atmosphere, some kind of medium. In our case, traveling through the air. And it's set up by vibrations, like a vibrating guitar string, for example. It's going to produce sound. And the frequency of the waves, how many waves per minute or per second, is going to establish the pitch. So high frequency is a high pitch. Low frequency gives you a lower pitch. And then the amplitude, like how big the waves are versus how small, is going to translate into volume. So the higher the amplitude, the louder the sound. And we measure that in decibels. Okay, so sound waves are what are going to stimulate the hearing receptors. So here's the ear with all the parts. The external ear begins with the auricle or pinna. So that's the part of our ear we see, and that helps direct these sound waves into the ear. And then the sound waves travel along the external auditory canal. You might remember something similar to this when we covered the skull, because we did see on the skull the external auditory meatus, which would have been here. That's the opening in the temporal bone. Because your temporal bone houses the ear. So you could see that on the skull, that region. Anyway, the external auditory canal carries the sound waves along the canal, where it will then impinge upon the tympanic membrane, commonly called the eardrum, but this is an anatomy class, so you need to learn the name of it, tympanic membrane, and it will cause it to vibrate. And the frequency it vibrates will match the sound waves. So if it's a high-pitched sound, it will vibrate with a high frequency. Low-pitched sound will vibrate more slowly. Okay, so that's the external ear, that part. The middle ear is shown in here, in this region. It has 3 little bones in it that you did not have to learn when we did the skeletal system, but now you need to learn their names. They're called the malleus is the first one. And then it articulates with the incus is the second one, and the third one looks like a little stirrup on a horse's saddle. It's called the stapes or stapes. People say it both ways. You might have learned these in a previous class with their common name: hammer, anvil, and stirrup, but again, this is anatomy class, so you need to learn their correct names: Malleus, Incus, Stapes. And they are in an air-filled chamber. This is the middle ear cavity within the temporal bone in here. And then notice there is a tube connecting the middle ear cavity to your nasopharynx, the upper part of your throat, and that is called the auditory tube or eustachian tube. And it helps equalize air pressure on either side of your tympanic membrane, because there's air in here in the middle ear, and there's of course air out here in the external auditory canal, and if the pressure is too different, it can put pressure on your tympanic membrane, and it can even be quite painful. If you've ever been, for example, a plane where the cabin's not pressurized properly, it can be very painful for your tympanic membrane. So having this auditory tube helps you equalize air pressure on either side of your eardrum. That's why when you're, for example, taking off in a plane, a lot of people will chew gum, or if you yawn or open your mouth wide, it helps bring air up into this chamber that will have the same pressure as the external pressure. Now, there's not supposed to be fluid in here, but if you do have a middle ear infection, then the auditory tube can help drain that away. But it's also because of it that you sometimes get middle ear infections, because pathogens can go up this tube into your middle ear and cause an infection. It's more common in children because they have shorter auditory tubes and they're straighter. As we get older, of course, they get longer and a little more convoluted, so it's harder for bacteria, for example, to go into your middle ear cavity. Okay, so that's the middle ear, and then this third part is your inner ear, which contains the cochlea that looks like a snail shell. That's where your hearing receptors are. And then the rest of your inner ear, which has these three semicircular rings called semicircular canals, that part, and then this region in here where I put the red X, and all of those parts of your inner ear contain equilibrium balance receptors. So again, your ear is a dual receptor for balance and hearing. Okay, so here's some pictures that just show these parts a little more clearly. Here's the external ear with the auricle, the external auditory canal, and the tympanic membrane in blue. So again, sound waves come in, travel down the external auditory canal, and hit your eardrum tympanic membrane and cause it to vibrate. Okay, now next, let's look at the middle ear. Here we see the three ossicles, three little bones. These are the tiniest bones in the body. This is the air-filled middle ear cavity and the eustachian tube. So when the tympanic membrane vibrates, notice that that's going to make your malleus vibrate because it articulates with the tympanic membrane. And then when the malleus vibrates, that makes the second one vibrate, the incus, because the incus articulates with the malleus, so they're meeting with each other. They're touching each other, so it will vibrate. And then when the incus vibrates, that makes the stapes vibrate. So your auditory ossicles, ossicle means little bones, your auditory ossicles help transmit the vibration caused by the sound waves vibrating your tympanic membrane. It helps transmit that vibration over to your inner ear where your receptors are. So we'll look at that. If you're exposed to a really loud noise, then we do have a muscle not shown here that pulls the malleus away from the auditory tube, or from your tympanic membrane rather, to help lessen the amplitude of that vibration. But you know, that's going to be, it's not always protective because this is in response to a loud noise. So if there's repeated loud noises, it can help for subsequent noise. But for that initial loud sound, you know, you're going to have you're going to accost the inner ear with a very large stimulus, which can be damaging to your ear. Okay, and then here's, oh, well, this is a close-up of the middle ear in color. So here we see, you can see better how vibrating your tympanic membrane will cause these 3 ear ossicles to vibrate. Okay, so let's look at the inner ear. So here's the inner ear. Try to picture this as, it's not really, although it looks like it's something you could take out of the ear and hold in your hand, but it's really best to think of this as a, this is within your temporal bone. So this is like the shape of the cavity within your temporal bone, and then there are cells and fluid and so on in there. But so that's your inner ear, embedded completely within the temporal bone. Here's the cochlea, the snail, where the hearing receptors are, and then the rest of it, the 3 semicircular canals, or here they're calling them ducts, and this middle portion called the vestibule, which is subdivided into utricle and saccule. Those have your equilibrium receptors, as I already mentioned. So let's look at few more pictures, although you see there's lots of detail that we could go into in the inner ear, but we are not. So I am simplifying the inner ear in this lecture, simplifying it quite a bit. But let's just look at the picture for a moment. So you could kind of think of your inner ear as like a tire with an inner tube inside. So oops. So there's the bony labyrinth, they call it, and in that space in there is fluid, they call it perilymph, and then it's like there's an inner tube inside of that called the membranous labyrinth, and it also has fluid inside called endolymph. Okay, well, I'm not going to be really asking you about what the fluid is called in the different regions so much as you do need to understand, though, that your inner ear has fluid in it, and because this is going to play a key role in how you stimulate your hearing and your balance receptors. So I do want you to know there's fluid in here, in the inner ear. Okay, and also let's look at this area right here called the oval window. It's a little hard to visualize this, but the oval window is between your middle ear and your inner ear. And actually your stapes, which you're not seeing in this picture, oh, well, here it is up here. So remember the stapes? It looks like a little stirrup. If you look at the base of the stapes, it's oval shaped. And your stapes fits up right against the oval window. So the base of the stapes, you know, covers this oval window, which you could think of as like a thin, kind of flexible window. And on the other side of it is fluid. Okay, so this is going to be important for how this works. So remember that where the stapes is, there's air in that space. But on the other side of the stapes, there's fluid in the inner ear. And what's between the two is the oval window. So we'll come back to that. That'll be important when we look at how this all works. So in this picture, we do see the stapes in the oval window. So there you see the stapes. Behind it or under it would be the oval window. Okay, so Also in this picture, I want to show you over here, if you cut open the cochlea, if you cut it open, you see that inside there's some cavities. So these gray areas are cavities filled with fluid, and then you see this tissue in between. I know this is very low resolution here, but you see that pink bar there. That's where your hearing receptors are. They're in there. So we're going to look at that. So what we're looking at here now is we are in the cochlea. So this is the the cochlea sectioned. Here you see a cavity above and a cavity below. This region where we have cells, this area is called the organ of Corti. And like I say, it contains your hearing receptors. So let's follow the path of the sound waves and how that eventually gets transduced into nerve impulses. In other words, how do we convert sound waves coming into our ear into nerve impulses that we can carry to our brain through the 8th cranial nerve, the vestibulocochlear nerve. Remember, that's cranial nerve number 8. Okay, so this is a close-up of looking in the cochlea. So this white space above and below is going to have fluid in it. This is fluid. So, and then here is the organ of Corti. So these yellow cells called hair cells, those are your actual hearing receptors. Like taste, the actual receptors here are not neurons. They are specialized epithelial-derived cells that are modified epithelial cells that serve as the actual receptor, in that they're hairs, which are kind of, you could think of them as sort of like dendrites. Their hairs are going to actually be stimulated, and they're going to have ion gates, and then that will open to cause the graded potential. So the graded potentials are triggered in the hair cells. We'll look at how in a moment. And then like in your taste buds, you have to have real sensory neurons, with axons that will synapse with the hair cells. And if those graded potentials in the hair cells are strong enough, then they can pass those on and trigger action potentials in the sensory neurons that will carry the nerve impulses away from your ear in your 8th cranial nerve. So hair cells, again, are not sensory neurons. They are specialized receptor cells that synapse with sensory neurons. Okay. There's also, in the organ of Corti, there's some supporting cells. Those are shown in orange. The support cells with their hair cells sit on a thin membrane called the basilar membrane. The basilar membrane. And remember, below that is fluid. And then hovering above the hairs of the hair cell is the tectorial membrane. And it's kind of a flimsy little membrane that's hovering above. Okay, and so that's kind of the setup for, we can now talk about how these hair cells get stimulated. So what happens is that when sound waves enter your ear, they, as we already talked about, they vibrate your tympanic membrane and then your 3 ear ossicles. And when your stapes vibrates, that vibrates the oval window, because remember, the stapes butts to the oval window, so the oval window vibrates, and that causes fluid waves in the cochlea. So the fluid out here is you're going to get fluid waves because the oval window is vibrating. And when these fluid waves are produced, then this tectoral membrane is going to kind of bounce up and down, and it's going to bend the hairs on the hair receptor cells. It's going to bend these hairs. You can kind of see how that would happen, right? If this tectoral membrane's flapping up and down or bouncing up and down, it's going to bend these hairs. And these so-called hairs, again, have ion gates in them. And as we've talked about many times before, ion gates that lead to either graded or action potentials must have some kind of stimulus that opens them. And in motor and interneurons, those are neurotransmitters doing that job. But for sensory neurons, it's something else. In this case, what's opening the ion gates is the bending of the hairs. So these ion gates are mechanically controlled. Remember, we talked about this in a previous lecture, the different kinds of stimuli for sensory neurons. Photons in your eye. Well, here it's these are mechanoreceptors. The physical deformation of the gate will open it. So hair cells are in that regard like touch and pressure receptors. They're mechanoreceptors. So you see why the fluid waves are going to cause these ion gates to open as things start bouncing around and the hairs get bent. Okay, and so ion gates open, you get depolarization, say, that's passed on to the actual sensory neurons, and again, if strong enough, it will cause action potentials that they will carry away from your ear to your cerebral cortex, and we'll look at that shortly. This is what it looks like under the microscope. So this is a slide you might see on your lab practical. So here are the hair cells, and then see this tectoral membrane hovering above it. So just imagine that bouncing around, because remember, out here this is fluid. Okay, so here we see the cochlea and the cochlear branch of the 8th cranial nerve carrying out impulses away. Now, I don't want to leave out the balance receptors, but we aren't going to go into it in as much detail. But I'll say this about the balance receptors. You do need to know they're in the other part of the ear. I think I already went over that pretty well. The 3 semicircular canals and this region here called the vestibule. So that's where your equilibrium receptors are. And they work actually quite similarly to the hearing receptors in that the receptor cells are also hair cells, specialized epithelial hair cells, whose hairs are bent by fluid movement, which causes action potentials which can be carried away through the vestibular branch of the 8th cranial nerve, and then they merge to form the vestibulocochlear nerve. So you see why it has this long name. I mean, back when I learned this 8th cranial nerve, they called it the auditory nerve. It was a lot easier name, but not as correct, really, because it left out the vestibular part. Anyway, you can imagine, I think, these hair cells being bent by fluid movements when you do things like twirl around, or do a somersault, or when you accelerate in a car or on a roller coaster or something, and so spinning, moving around, acceleration, deceleration, all of those things are going to cause fluid waves in the inner ear, and that will stimulate those hair cells. So it's similar to hearing. It's just really the source of the fluid wave. Is it sound waves for the cochlea, or is it the body actually moving position for the other receptors, the vestibular part of the ear? Okay, so this shows the auditory pathway. So again, what that means is what is the path the action potentials take from the ear to their final destination. So let's look at the final destination for a minute. It's the primary auditory cortex of the cerebrum. Okay, this is something we already covered with the brain, but you definitely need to, you'll be asked about this. So remember that the impulses from your ear need to go to a particular part of your cerebral cortex to be processed so that we can hear. And we also talked about how the axons cross over, so impulses coming from your right ear go to your left primary auditory area and vice versa. So here's the pathway. We go from the hair cells, which begin the whole process with graded potentials, out the ear along the 8th cranial nerve, vestibulocochlear nerve, and then from there the action potentials go to the thalamus. And remember, that's always the case for taste, for vision, and here for sound, but not for smell. So they go to the thalamus, and then the thalamus directs it to the primary auditory area. So if we're just looking at action potentials, it would be vestibulocochlear nerve, then to the thalamus, and then to the primary auditory cortex. For vestibular information, there is a similar pathway going from the ear to the thalamus, but then to the vestibular area, which we did not talk about when we covered the brain, but it is sort of in the same region. It's like in this area up here. So it's a similar pathway in that you're going from the ear to the thalamus and then to the cortex. Okay, so that completes our lecture on the eye and the ear.